from now on is that these two sequences, whatever they are, have very low sequence identity and so present a very challenging problem. They're going to be into that twilight zone. And one of the exercises that you have in your tutorial is just to show that when you have very low sequence identity, that the Smith-Waterman-Needleman-Wunsch methods sort of start failing, and that you don't get very good alignments, and that you really should go over and use some type of profiles. So I think the exercise will make that very clear. And as I said, one of the things that helps you in thank you. In developing these profiles is to go back and make use of evolution. And one of the things you do is you have to go back and start reading Carl Voss's papers, and when he first introduced this universal tree here, right? And first I should tell you that what this universal tree attempted to do was first to give you hey, thanks, Felix. I got lots of water. One of the things that this universal tree tried to do, if you look, these are mostly organisms that are written down here, tried to tell you how the organisms are organized along the three domains of life, the bacteria, the archaea, and the eukarya. But as I said, he did this analysis working on a particular set of sequences, the ribosomal RNA sequences, because, you know, that's something that's, you know, fundamental. This information processing apparatus has got to be there in all the domains of life. And it gave you this particular structure of how things should be related. So there was some, you know, root of this universal tree, and then you started getting differentiation into these various domains of life. Now, the interesting thing is if I switch now from ribosomal sequences to, say, other molecules, do I get the same molecular phylogenetic tree? Like, so, for example, if I look at other sequences involved in the information processing, do I? In general, are the archaea and the eukarya more closely related than, say, the bacteria? And the answer is, for certain components of the cell, yes, that's true. If you're looking for some general rules, again, I encourage you to go read Carl's paper on the origin of the universal tree, and then his more recent ones in 2000. So if you start looking into things involved in more metabolic pathways, you'll start seeing variations of these themes, and those variations are very important. And they tell you a little bit about evolution of that particular protein. And the main cause of that variation that you're going to see, they give it a name called horizontal gene transfer. So let's call this picture that Carl first gave us is the canonical picture based on the ribosomal RNA sequences. But you'll see that sometimes what has happened is that a protein suddenly, if you look into this branch, which was a eukarya, either it's not even there, or it'll be totally of the kind that of the sequence that came, say, from the bacteria or the archaea. There has been some transfer of genes from one domain into another, a species in an organism in another domain. And so you can see terms that people use in evolution called full canonical, basal canonical, or just non-canonical. There's been so much transfer that takes place, you really can't sort of draw back its genealogy. Where did it really come from? So if you're trying to form a profile of a particular molecular, a particular protein, you've really got to decide, or have a bit of a feel beforehand, where am I in here, right? Should I, maybe the simplest notion you would have, let's try to build profiles out of related sequences. First, I'll build up all the bacteria ones, and then I'll pull out all the sequences from the archaeas, and there are lots of organisms in the archaeas, and all the ones from the eukarya. And then maybe I'll try to get a feel for the amount of variation I should observe, and if there are any patterns at all, maybe I compare them, all three. There can also be other cases where, maybe one whole branch will not even be there, right? And again, you have to get a feel for the molecular phylogenetic tree and in trying to develop these profiles. So that turns us to looking at a particular protein. And the one we've chosen for your tutorial are these aminoacyl-tRNA synthetases. And again, if you listen to Carl, he really likes these because he says perhaps better than any other molecules in the cell, they epitomize the current situation and help to understand, and I put in here, the effects of horizontal gene transfer. So you're going to see that some of them, and I'll explain in just a moment what do these aminoacyl-tRNA synthetases do. They're responsible for loading the amino acids onto the tRNA that is used to produce the proteins in the ribosome. So here, if I'd had time, was going to come a nice picture of the standard dogma of molecular biology. Forgot to do it. But I think you all know, in generally, the way it works is you have a DNA sequence, you get it copied to an RNA sequence, or it can copy to itself. And then from the RNA sequence, after it matures, it goes to the ribosome, and then you have to start bringing in one amino acid after the other to convert the DNA code, the genetic code. And there's like 3 bases that define a particular amino acid. And there are, for any one particular amino acid, often more than just one codon pair. There's a lot of degeneracies, they're called. So there'll be several triplets that will correspond to a particular amino acid. And those guys that help transfer it are these little tRNAs. They have to get charged, as they say, or get loaded with the particular amino acid and bring it to the ribosome so you can start making the proteins. So they're sort of interesting guys in that they connect with the information processing to sort of the outside world. And you see from this next study that was actually done by one of my students, Patrick O'Donoghue, and we've just been invited to submit this as a review to where Carl first submitted his analysis of these sequences. You're going to see every pattern in there. You're going to see for some of them, they're going to have a canonical pattern, some of them are going to have a basal canonical pattern, and some of them will have no pattern discernible at all. So they're at all stages of horizontal gene transfer and of evolution. Here's a typical one that we pulled out of the national databases, NCBI. The reason I think, we did this from them, is they also have the ability, and I think it's sort of not so visible down here, it's color-coded by domains. So the entire synthetase is actually very large. It interacts with the tRNA. As I said, one of the things it has to do, and it's a little hard, I can't see it myself, just a second. There's an acceptor arm of the tRNA where you have to load up the amino acid. There's also a portion of it to know, sort of, there's a lot of communication that's going on, like how do they know this protein, which amino acid should it be loading, right? You can imagine it's docked in this little space here, and a tryptophan is certainly different from alanine. So there's going to be some differences maybe here in the docking site and the amino acids that are there. And in fact, if you to look at all of them, say for all the 20 amino acids, there's less than 10%, around 10 to 15% sequence identity, say within the core of this molecule, where it docks the amino acid and puts it onto the receptor arm. But equally important is this green area up here, which is the anticodon region. Here is the anticodon region that's on the tRNA, that special triplet that codes for it. There's a part of the protein that interacts also with that region. This region is also what is then read over in the ribosome, and it has to match its mirror image on the messenger RNA, and then it loads on and starts building the proteins. Now, what you're going to do in your study here, we're not going to look at the whole synthetase, because it turns out even in some of them, this portion is missing. And the only thing that's really strictly common to all the synthetases for all the 20 amino acids is this region we call the catalytic domain. And this is sort of this reddish color here. And that's what you'll be studying and looking at the variations that are there, both within a class, say like all the ones that load aspartic acid from all 3 domains of life, plus comparing those to ones that say load histidine, something totally different. Again, from several different domains of life. And now I just want to show you 1 or 2 pictures out of a paper that Carl Vos and Gary Olsen and Sol wrote in about 2000, when they did sort of the ultimate analysis of all the tRNA synthetase sequences. These are all the sequences from all the organisms that they had available at that time, and that load a particular amino acid, in this case leucine, onto the tRNA. And if you look, I wrote it up here for you, that if you were to look at the names here and the coloring, you would notice this obeys what he calls the canonical tree, in that here are all the eukarya, and they should be closely related to the archaea. And there's Carl's favorite one, that's Yanashai. I will also have you guys look at that one, because not only is it his favorite, he was the first person to publish the sequence on this and raise many of the issues about evolution and the genetic code. And I think we also have you look at that article, because it's really very historically extremely important. So, and again, I think you can see that the archaea and the eukarya are grouped closer together than all the bacteria ones, okay, for leucine. Let's go now to another synthetase that loads not leucine, but aspartic acid. And again, you start seeing some variations in here. You know, in general, it's like, again, these are the eukarya, are more closely related to the archaea, but there is a guy who sort of stands out, who should be, people call him a bacteria, but he's up here, and if you look at the sequence, it's actually much closer to these guys. So this is an example of where this thing, horizontal gene transfer, then took place, and took that particular one out of, and made it more similar into this class, into this, domain or genre. Now, let's take a look at what these tRNA synthetase catalytic domains look like. And in general, there are going to be 2 different classes of them. Usually, class 1, there's like 20 amino acids, and roughly you can say class 1 codes for 10 of them, and class 2 codes for the other 10. On occasion, there are synthetases that actually, in this case, there's a class 1 and a class 2, both for lysine. And I should also say, I keep saying there are 20 amino acids. I think we all know now that they're actually, the accepted number is like 22, and one of them, the newest one, this pyrolysine, was discovered last year. And in fact, sort of the homework assignment you did on generating this charm potentials to be able to study this sort of non-standard amino acid, that's a watered-down version of a problem we gave in one of the courses here, which is to try to develop new charm force field parameters for this pyrolysine to be able to study it in molecular dynamics simulations. These are the 2 structures for the class 1 and the class 2. I think you can see they are really different structures. This one is a Rossmann fold. This one has a novel fold. There's like, besides the class 2, there's I think only one other protein out there that has a similar structure to this. So it's, they really don't look like they should be related to one another, but they actually do the exact same thing. They both load amino acids, in this case the same amino acids, onto tRNA. Now, let's look at the variation in the structures that we see. So we can start thinking about developing a profile of the structures. And what we've done here for the class 1 as well as for the class 2, we've taken all the tRNA synthetases from some representative set. They all load different amino acids, and we piled them on top of one another for the class 1, and then taken the other 10 and piled them on top of one another over here. And this is the same picture in all these panels, just different views of it, but I think you can really see very easily, and you'll use this VMD to do this, and these are some of these nice scripts that poor Barry was up all night working on to help you analyze just exactly what is the core and what are really the insertions and deletions that have taken place as it evolved to allow you to look at, as the thing has evolved, to be able to identify and load on aspartic acid, say, versus lysine or something like that. And I think you can see very easily, and this suggests some early protein that maybe was the ancestor of all these guys. And we did the same thing over here into the class 2s. Now, you can take those sequences, and this is part of Patrick's work. You know, what we're interested in was looking at the evolution of structure to go along with the evolution of sequence analysis that Carl had done. And can you also develop phylogenetic tree analysis, very similar to what he had done? And the answer is yes. And the reason this is very important is there's sort of like regions of sequence identity and structure identity that are complementary to both approaches, and then there are regions where you can only work with really structure and other areas where you can only work with sequence. And again, we'll try to make that clear to you and you do your homework, that when you get down to very low sequence identity, that you are really best helped by trying to develop your phylogenetic analysis of doing just the structures. In the regions where they can overlap, you can do both, and then that helps you get an idea about these things that are related by sequence. What does that mean in terms of the structure? What insertions were allowed, and where, and why, and what's in contact with what? How much did it change the core? How much did it change its interaction with the RNA? But as you move out to this region, which is very fun in some ways, you're moving towards greater and greater structural identity, structural similarity here. You sort of are fading off in your ability to make really strong statements about the ordering of the structures, and in part because of the artifacts that are in the structures themselves. They've been crystallized by different techniques. Some of them have contained the RNA, some of them contain the amino acid. So over here, where they become structurally very close to one another, if you want the exact details of this, it's best to go to the sequence, because that's where it really functions well. It has high sequence identity, and then you can check this ordering. Now, this just took everything that was available to us in the PDB database, and as I said, it's very important when you're working in this field that you work with a representative set, because if you're going to learn anything from it, whether it's learn interactions for an energy function or learn interactions to develop a profile, you have to know sort of, do you have really representative members in there, or do you have 10 copies of something that's practically the same? So one of the key things that came out of Patrick's work, which is he developed a new technique to really define what we mean by non-redundant sequences and or structures, which was really being able to allow us to push this whole analysis of molecular phylogenetic, the trees down to lower values, and also enable us to work with a smaller set of sequences in doing in any particular study. In fact, I can show you, and these numbers here sort of indicate which ones are most representative. You can go from that picture down to this one, which is now makes everything much easier to do, right, and to work with. Now, that was for the class 2s. I just want to show you he's done also the analysis for class 1s. You can just show these all day, and if you're hanging out with molecular evolutionary guys, they just love it. Everybody else goes, my God, what is this, right? And so we'll try not to bother you with too many of them. The only thing that we will have you do is look at a sub-branch or subsection of this representative tree, and in fact, we'll go back one, and we'll put in a few that are, say, these are all, if you were to look maybe out of certain bacterias. So if you then go and do a phylogenetic tree based on sequences, you should see some things are very closely related and some are more distantly related to one another. And in fact, you'll see that there seems to be some mixing in of these, or there's some way the class 2 lysines are somehow related to these aspartic acids. You know, what's going on there exactly? Can I look at that evolutionary history? Can I try to understand this better from doing this? And the answer is yes. And if you go back and try to draw a molecular phylogenetic tree from that, you can show from looking at that more detailed picture I did that somehow they were all related, had a common ancestor here. Then there started to be this branching into these domains. Sometime or another, this one, lysine, then underwent horizontal gene transfer over onto the eukarya branch, so then it became much more similar to the eukaryotic synthetases that load aspartic acid. But the funny thing is, you don't see K2 popping up here at all. And in fact, just to check, I asked Patrick again, sure we didn't leave it out? And the real answer is, there aren't any in the archaea world of the class 2 synthetases for lysine. So as I said, you can then start using this to make entire scenarios about the evolution of which amino acid came first and how are they related to one another. And the reason that's ultimately also helpful to you is it gives you an idea maybe how you should think about grouping them together in any structure prediction or any treatment that you do or analysis of a sequence. Now, it's not possible for me to go into all of that, and a lot of it is just still being thought out in my own group of how to maximize this. But it is clearly this is the wave of the future and how things will develop and how you can sort of use evolution to help you with this analysis. Now this picture is somewhat incorrect, but I was hoping then to just summarize the things that we've learned. Like this is what we've got from Carl, the fact that certain of them amino acids appear to be can be broken up into canonical basal particles, basal canonical patterns, or such strong horizontal transfer that you can't recognize their origins anymore. That gave rise, I should have had the same tree I had here before, but this was an older slide and I just didn't have time to redraw it. So there was the sequence things that Carl had done, and now should come in slowly the structural world. And so now we can look at every change of the sequence, how it corresponds to what's going on in structure, and that's a very, very powerful tool. Not only out of looking at what those changes were, we can get an idea about those distributions of gaps, what they look like, fit them to different functions, and this is how we improve on those threading algorithms now, is we have much more reliable data of what should be expected and how to treat it, and then you can fit them to new functions, and those will go into anybody's, including our own, threading algorithm. I should point out, and I just felt like it would be an overload to show you more of these pictures. We did it first with these tRNA synthetases, these aminoacyl-tRNA synthetases, because from an evolutionary, from a pure basic research point of view, those were the most interesting ones to do, because they help sort of make more firm this whole story of evolution and know how to use it. And there were a lot of reasons, as I said, more from a basic and pure research considerations. But it's obvious that where the real strength of this whole approach comes in is you can do this analysis on any of the standard folds that are out there. And again, as we do more and more of these genomic sequences are done, we have the structures that have been determined, we're seeing that there are some really important alpha-beta folds that need to be considered, and exactly the same treatment that one did for the synthetases, one can apply to those other ones, and one is doing this, and it really helps with the interpretation of looking at how function has evolved in these structures. And at least in chemistry, there's this whole field called chemical evolution, where people are trying to change one enzyme to do the work of another, and this gives you an idea of what those changes are that you'd have to make and be able to do this in the easiest way, sort of learn the lessons of evolution. And in fact, this just shows the results. We've already done a number of these out of all the basic folds, the alpha helical folds, the beta sheet folds, the alpha beta folds. We've done our analysis. We can get gaps from them. Sort of interesting on certain levels that you see sort of similar types of gaps and structures coming in, which is reassuring. That means we don't have to keep redoing this analysis for all the basic families, that maybe we can work with a cruder one initially. Okay, I thought now we can make a longer break, particularly since Klaus is out of the room, and come back, and then we'll start talking about some of the algorithms that one used. Right now I'm just showing you the results. We'll get into a little bit more of the details of how one did those structure alignments, what is out there, and then we'll get into the web tools and discuss a bit your tutorial for the afternoon. Okay, thank you very much.